Okay, so here is part two of the of chapter 26. So we're going to pick up on what makes bacterial cells pathogenic. So what makes them cause diseases? Well, one thing is virulence. So virulence is the ability to cause diseases. It's also heritable and a variable trait. Some species have both pathogenic virulent strains and harmless strains. For instance, E. coli, it has genomes of pathogenic strains that are larger because they have acquired virulence genes. For example, a gene that codes for a protein toxin, okay, E. coli, the strain E. coli 0157 H7 is pathogenic to humans. Okay. Some pathogenic bacteria produce resistant endospores. An endospore is a tough, thick-walled, dormant structure that's formed during the times of environmental stress. So in this form, the endospore is within the cell, within the bacterial cell, but it has a copy of the cell's DNA, RNA, proteins, and enzymes. So in this case, when the metabolic activity will slow down, and then the cell will break apart. Essentially, it'll break down, and all that will be left is the endospore. And so the endospore allows it to go. It's almost like a dormant phase that allows it to be resistant to things like UV radiation, high temperatures, or antibiotics. And then when favorable conditions return, then the cell resumes to grow. So antibiotics are molecules that kill bacteria or stop them from growing. They're produced naturally by some soil bacteria and fungi. Um, they were created in the 1920s, like 1928, okay, and they were widely used in the 1940s. So because they were widely used um, in clinics and also in animal feed, that led to the evolution of drug-resistant strains of pathogenic bacteria. Biofilms are bacterial colonies enmeshed in a polysaccharide-rich matrix that shield bacteria from antibiotics. Okay, so a biofilm is just like it's like a membrane surrounding the bacteria. Bioremediation is when you use bacteria and archaea to clean up um, sites that are polluted. So it could be like water pollutants that are toxic to eukaryotes. You know, they the water pollutants they also do not dissolve in water and they can accumulate in sediments. But naturally existing populations of bacteria and archaea can grow in spills and essentially break down the toxins. Bioremediation uses two complementary strategies. Fertilizing, which contaminated sites uh, are encouraged to grow with existing bacteria and archaea, and also seeding or adding specific species of bacteria and archaea to the sites. All right, so how do biologists study bacteria and archaea? Well, they use several methods. The first method is enrichment cultures. So that's when you grow bacteria under specific conditions. Then there's metagenomics. There's investigating the human microbiome, and there's molecular phylogenetics. So the enrichment culture most describes species discovered in controlled conditions in a laboratory. So they isolate large populations of cells that grow under specific conditions, and this led to the discovery of the thermophiles, the heat lovers. So here's an experiment where they wanted to see if um, bacteria live a mile below the Earth's surface. So they wanted to see if bacteria was capable of cellular respiration deep below the Earth's surface by using hydrogen as um, an electron donor and iron as an electron acceptor. So they prepared a culture with a lot of hydrogen and iron and raised the temperature to 45 degrees. Then they added rock and fluid from samples they got from drilling at the depths of about 1,000 meters below the Earth's surface. And they found that some species actually did grow in the enrichment culture. Using metagenomics. So metagenomics, also known as environmental sequencing, identifies species and biochemical pathways by comparing DNA sequences with those of known genes. Then they identify and characterize organisms never seen. They use in combination. They're used in combination with direct sequencing, which um, isolates and sequences a specific gene from organisms found in a specific habitat to understand the whole diversity. So here you're looking at the human body, and so there's different parts of the human body that serve as microbiomes. Okay, as you can see, the mouth is one, the male gut is another. Oh, sorry, not the male. There's both male and female on both sides. So we have the mouth, the the intestines, and also hands. So those all serve as microbiomes. So a microbiome is a community of microbes that grow naturally in parts of the body. Humans have a diverse ecosystem of symbiotic prokaryotes. So, like I mentioned before, not all of the prokaryotes are like, including bacteria and archaea, not all of them are bad. So some of them actually help you. So you have a lot of bacteria that actually helps you, live. The gut microbiome that has about 100 trillion bacteria and archaea. Some microbiomes, some microbes, they can make us sick, but like I said, we also depend on a lot of them to stay healthy. And also changes in the gut microbiome have been linked to inflammatory bowel disease and obesity. So that's why it's important to have a diverse diet. So they say that when you consume a lot of vegetables or fruit, you actually help support your healthy bacteria. Or like fermented foods can also have a lot of good bacteria. So some of the most useful phylogenetic trees for domains bacteria and archaea have been based on studies of RNA. So they use RNA sequences to see um, which of the domains are how similar the domains are. So in 1960s, Carl Woese and his colleagues, they compared 16S and 18S RNA molecules from a wide array of species. And what they came up with after analyzing the DNA sequences was the tree of life. So I think I've already covered this in lab, but this shows you the relationships among bacteria, archaea, and eukarya. So based on those RNA sequences, they found that archaea was more closely related to eukarya than it was to bacteria. So these two are considered sister taxa on the phylogenetic tree because they have a more recent common ancestor here. And here's just an example of the diversity that you can find in bacteria. Okay. Not as much as in archaea, but there's still some diversity here. Okay, so themes. So we're gonna leave this for the next video. Okay, so we're gonna continue with themes that occur in the diversification of bacteria and archaea.